Jim. My name is Milan Radovich. I'm the Senior Vice President and Chief Scientific Officer at Caris Life Sciences. I oversee much of our R&D, translational research, drug development, and our clinical laboratories. I've been at Caris about 20 months. Prior to being at Caris, I was an associate professor and vice president for genomic oncology at the IU Simon Comprehensive Cancer Center, where I led a clinical precision medicine program and also did research in liquid biopsy, drug development, as well as MRD. At Caris Life Sciences, we pride ourselves on using cutting-edge technologies to really figure out what makes each patient's cancer tick. And we do this by using whole exome and whole transcriptome sequencing, meaning we interrogate all 22,000 genes in the genome, both at the DNA level and at the RNA level, trying to figure out what are all the mistakes in the cancer blueprint that both may have led to the cause of that cancer, but more importantly, what therapies we could find to stop the disease. As you may imagine from cancer therapy, cancer is a disease of DNA. It's caused when there's mutations that occur in the DNA blueprint that causes a normal cell to go haywire. And what we've been able to do in recent years through advanced cutting-edge technologies is scan the DNA of cancers, figure out what all those mistakes are, and match the right therapy to this. We call this concept precision medicine, tailoring therapy to the individual makeup of each patient's tumor. Now, the reason we look at all genes in the genome is for two major reasons. One, we don't want to leave any stone unturned in identifying a potential actionable alteration for a patient. So if there is a drug out there, whether it's FDA approved or on clinical trials, we want to be able to find that drug for that patient. We want to have the peace of mind that we have given that patient every single chance to find a drug for their cancer. The second important piece of why we go comprehensive is also for versatility. What we know is that new markers come out almost every week in the literature and in clinical trials. We don't want to be restricted to a 300 genes or so panel where if a new marker comes out, it's not present in our panel, we have to revamp and redesign our particular gene panel. What we love about our approach is that if a new marker comes out, we already detect it. And this actually happens relatively commonly at Caris, where maybe at one point a particular drug wasn't available for a patient, a new marker and a new drug comes out, let's say several months later, we'll actually go back and look at all our previous cases and try to identify patients who are positive for that particular marker. Again, wanting to have that particular versatility. With this sequencing data, we've amassed now almost 500,000 patients of sequencing data. It's the world's largest clinico-genomic data set. And we don't keep this data to ourselves. We're really passionate about advancing research in the field of precision medicine. So we have banded together now with 85 cancer centers across the country and the world through our Precision Oncology Alliance. This is comprised of many individuals who are experts in medical oncology and bioinformatics, cancer genomics, pathology, radiology, and other specialties, where we band together to utilize our data set to advance research. The POA is actually separated into a variety of disease groups, so focused on different tumor types and also different specialties like pathology, radiation oncology. We also have a fantastic healthcare disparities council as well. And it is a very academically productive group. Actually, we are presenting 45 abstracts here at ASCO from this research. Our academic partners, as you can imagine, utilize this data quite extensively. We also bring to bear a very significantly large team inside of Caris that works with our academic investigators that perform the analysis together and develop the posters and write the subsequent manuscripts as well. So again, we really wanted to make sure that not only are we giving each individual patient that we take care of the best chance for fighting their cancer, we want to help the patient in the future utilizing this data to its maximum extent possible to advance research in this disease.